So I have gone through the glomerulus. I have traveled through the proximal convoluted tubule, traveled through the loop of Henle, or the nephron loop, sorry, traveled through the distal convoluted tubule, gone through the collecting duct, and formed urine all in that process. But there's one last thing we got to do before we ultimately leave and talk about urine. We have to talk about forming a concentration gradient, okay? Concentration gradient is what is formed deep in the medulla. The nephron loop of the juxtaglomerular nephrons are the guys and gals that are primarily involved in this process of forming a concentration gradient. It is this concentration gradient that allows us to maintain sodium levels, chloride ion levels, water levels. This concentration gradient is what it's going to allow us to maintain and hold on to stuff that we ultimately need to, or get rid of stuff that we ultimately do not need. It's referred to as a countercurrent multiplier system. That's what we're going to be describing here. All right. But it involves the nephron loop, and it involves the blood vessels that surround the nephron loop. The vasa recta. Sorry, brain fart. Okay. We are going to have, and I'll just stay over here. We have a descending nephron loop and an ascending nephron loop. And when we're talking about the countercurrent multiplier system, we are looking at this descending limb is a heck of a lot more permeable to water. As we get deeper and deeper and deeper into this renal medulla, we have a higher and higher concentration. 300 milliosmoles at the top of the medulla and 1200 milliosmoles down towards the bottom of, or the deeper portion of the renal medulla. What does everything like to maintain? Everything likes to maintain equilibrium, right? That's the goal of everything. We have sodium chloride in here. Let's just talk about that one. So sodium chloride and water are within this thing. It all got forced out in the glomerulus. Sodium chloride and water are traveling down in here, and as we get deeper down into the medulla, we come across a higher concentration in the surrounding tissues, in the interstitial space, or the interstitial tissue space, called the interstitium generally. The only way for that content within that tube to become more concentrated is for water to leave. So the descending limb of the nephron loop is highly permeable to water and impermeable to anything else. You can see as water leaves, it gets picked up, because if water left and just filled up the tissue space, you would assume it would just wash it out. It does not, okay? We pick up water here within this vasa recta. Vasa recta leaves, vasa recta flow is going in this direction, hence countercurrent. One this way, and it picks up that water and returns it to where the milliosmolarity is at equal with the contents of this fluid. As we make the turn here, nothing is really moving. Tubular fluid is a hell of a lot more concentrated. Again, I've pumped out near all the water. But now we go up this ascending nephron loop. Sorry. Now we go up this ascending nephron loop. And what are we doing? We are going from highly concentrated to less concentrated. So now what's going to happen? Well, the only way for this to become equal now, because as you can see, it's impermeable to water, is now I need to have the little bit of water that was in here. The only way to bring it back to around 300 milliosmoles, and it can get as low as 100 milliosmoles, is to be really permeable to salts. So the ascending nephron loop, ascending limb of the nephron loop, is highly permeable to sodium chloride. Sodium chloride leaves, gets picked up by the vasa recta here. And remember, the vasa recta is going down. This is going up. And as it descends back down, it becomes more concentrated. So let's go back here to the words. Right? Descending limb, permeable to water, impermeable to salts. Okay. Water moves from the tubular fluid to the interstitial fluid and ultimately will be picked up by the vasa recta. This allows the tissue to become concentrated. The ascending limb, like I said, is impermeable to water, but it's permeable to salt, so salts get pumped out. More concentrated the salts, the more is pumped out. And we saw that when we looked at regulating glomerular filtration rate. And we create this concentration gradient within the interstitial fluid. Okay, that's what's happening here. Water is being pumped out, becomes more concentrated, and as we head back up, gets to lesser concentration, we pump out NaCl. That's the countercurrent multiplier system. We also have a countercurrent exchange system, where the countercurrent multiplier system involved the loop of Henle, the nephron loop. The countercurrent exchange involves the vasa recta. And again, countercurrent tube going in opposite directions. Where the countercurrent multiplier system establishes the concentration gradient of the renal medulla, which is important for forming a more or less concentrated urine. The countercurrent exchange system helps to maintain that concentration gradient of 300 to 1200 milliosmoles. All that water that I talked about that got pumped out of the descending limb of the nephron loop gets picked up here. Okay. So here, as the blood comes down, because this is the vasa recta, okay, that blood gets, the blood, as it comes down, it has to be isoosmotic with the surrounding tissue. So it gets picked up, or it gets pumped out. NaCl gets pumped in. Where did NaCl come from? Right here. Okay, right here. NaCl is being pumped out. As we make the turn back up top here, where we're at 300 milliosmoles, the only way to make that back to normal is to get water to go back in it. Where'd that water come from? Poop, right there. Okay. That water that got pumped out through the descending limb goes back in there. Some salts leave as well, okay, to maintain that isoosmotic concentration gradient with the surrounding interstitial space. Again, this is what helps us to form a more or less concentrated urine. Because remember, this is 300 milliosmoles right here. Okay, but right here, it can be as low as 100 milliosmoles. Okay. Because remember, this is the distal convoluted tubule right here, right? And then what's it dump into? The collecting duct. And where's it going? It's going to travel right down that too. So can we affect the collecting duct and affect the concentration of it? Yes, we can. Antidiuretic hormone. It's going to help us to put more or less transport aquaporins to pump water in or pump water out. Okay. But this concentration gradient is utterly important. Okay. It helps us to maintain a concentration gradient, which allows us to maintain the ability to form a more or less concentrated urine, to form a more or less copious amount of watery urine. There's one other major thing that is involved with forming a concentration gradient, and that is urea. You don't urinate all the urea out. You actually recycle some of it. Because once the filtrate goes through the glomerulus, right, and becomes filtrate, goes to the proximal, the nephron loop, and the distal convoluted tubule. As it enters into that collecting duct, it's basically urine at this point. And it has urea within it, okay? As this urine now travels down the collecting duct, it's going to go through this concentrated medulla. A little bit of urea pumps out of that collecting duct to help maintain some of the osmolarity of the surrounding interstitium. If there's already a significant amount there because there's been a lot of urine formation going on, it will retain it. Okay? It'll retain more of it. And you produce more urine in the process. Urea helps in the concentrating of that renal medulla, of that interstitium. Okay? It's not just NaCl. The biggest ones to remember, the biggest ones to know is always the countercurrent systems. Okay? That countercurrent system could be a potential short answer question, but more than likely it's going to be multiple choice questions. Okay? Urea is just the other sister in the room that nobody pays attention to, but it helps in concentrating the urine just as much as NaCl. So if I was to sum this all up, after filtration, majority or most of the substances are reabsorbed or secreted. Okay? The nephron loop, the vasorecta, the urorecycling, they've helped us to establish this concentration gradient within the medulla, which is to say within the interstitium. This helps us to determine, it helps our body to determine how much antidiuretic hormone needs to be released. Do I need to release a lot of it? Do I need to release none of it? Very little of it? That's what that concentration gradient allows us to do, to form a more concentrated urine like I keep talking about. What controls that? Well, it's those cells that we made reference to way back at the beginning, principal cells, intercalated cells. Those are involved with, the process of reabsorbing hydrogen ions or carbon dioxide ions or secreting those things, okay? Urine is composed of water, dissolved substances, and waste products, and that's what's basically running through the collecting duct. Renal papillae, minor calyx, major calyx, renal sinus, ureters, urinary bladder, urethra, toilet.